Hello and welcome. In this video I will demonstrate how we can use an automated cell counter that automatically counts cells. This benchtop instrument is for easy cell and viability counts. It counts cells in as little as 30 seconds accurately and precisely using the standard Trypan Blue technique. Cell counting chamber slides. These are plastic, disposable, enclosed chambers that holds the sample in two separate chambers, A and B, for replicates. So our first step is to get our cell culture flask from the incubator and observe under the microscope. If it has almost 80% to 90% confluency, then we need to passage our cells. First of all, we need to aspirate off all the media from the cell culture flask and then wash the cells with 10 ml PBS or HBSS and rinse the cells in PBS or HBSS by gently swirling the flask. Now add appropriate volume of trypsin EDTA like 1 ml in T25, 2 ml in T75 and 3 ml in T175 flask to sufficiently cover surface area to detach the cells. Now incubate the cells for 3 to 5 minutes in the humidified carbon dioxide incubator. The incubation time depends upon the cell type that we are using, and you need to optimize it for your cell type. As we can see, all cells are detached from the flask. Now we need to neutralize the trypsin EDTA with warm FBS containing growth media. Now resuspend the cells by gently pipetting the cell suspension up and down 3 to 4 times and transfer into a 25 ml tube. And now centrifuge the cell suspension for 5 minutes at 900 to 1000 RPM at room temperature. As you can clearly see the cell pellet at the base of the centrifuge tube. Now remove the supernatant and resuspend the cell pellet with the pre-warmed fresh media. Resuspend the cells by gently pipetting the cells suspension up and down 3 to 4 times. Now take 20 microliter sample of cell suspension and transfer into a sterile pen drop tube. Now add 20 microliter of Trypan Blue into the pen drop tube and mix well. So finally, to begin with the cell counting, we will use our cell counting chamber slide and we will mix our Trypan Blue cell suspension mix, and we will add 10 microliter of Trypan Blue cell suspension mix and carefully pipette a drop of the cell suspension into the well of the counting chamber, allowing the capillary action to draw the sample in the counting chamber, as it only holds 10 microliter, so take care not to overfill the chamber. Well, after the insertion of cell counting chamber slide into this benchtop automated cell counter instrument, now we can see the cells on the screen, and after we'll press the count cells, it will give us the total number of cells and the percentage of the viable cells in just 30 seconds. So wait and watch. Well, our live cell count is 4.9 into 10 raised power 5 per ml and viability is 91%. And now if we want to seed 40,000 cells per ml, we'll use this formula and we have to add 0.081 ml, that is 81.6 microliter per ml will give us 40,000 cells per ml. Thank you for watching.